For those of you who've been looking for Season 1 of Solarpunk Presents, but haven't been able to find it, here it is. Originally, it was published as part of Solarpunk Magazine's Solarpunk Futures podcast. We're reposting our episodes on our site to make them easier for you to listen to if you didn't catch them the first time. We hope you enjoy them. Also, don't forget to support us by spreading the word about Solarpunk Presents, writing us a review, or subscribing to our Patreon at patreon.com/solarpunkpresents. Hello and welcome to Solarpunk Presents, the companion podcast to Solarpunk Futures. Hosted by Solarpunk Magazine nonfiction editors extraordinaire Ariel, Kroon, and Christina de la Rocha. Ariel and I will be using this companion podcast to Solarpunk Futures to explore solarpunk goings-on in the world today. We'll be interviewing all sorts of interesting people who are doing work in the here and now that will help us get to a solarpunk future. And we'll be talking to each other about the visions of a sustainable, equitable future integral to solarpunk, and about issues we're curious about within the movement or genre, or whatever it is you want to call solarpunk. And now for this week's episode. Do you think the deep sea is dark? Totally, utterly, incorruptibly dark. In that case, welcome to episode 7 of Solarpunk Presents, inspired by the lunarpunk issue of Solarpunk magazine that came out earlier this month, and in which we learn that actually almost everything living in the deep sea glows, flashes, and/or has headlights. We'll be talking today with Dr. Steve Haddock, a senior scientist at the Monterey Bay Aquarium Research Institute, MBARI for short, in Moss Landing, California. He's the biggest bigwig in the field of deep-sea bioluminescence. Hi, Steve.
Hey, Christina.
Thank you so much for talking to us about bioluminescence, as you are one of the world's leading experts on deep-sea bioluminescence. Sure. A fun place to start would be with how bioluminescence works. What happens when an organism lights up? Or how does that happen?
Yeah, I mean, in some cases, we're still trying to figure it out. But in general, bioluminescence is when an animal makes light, or an organism, actually. Doesn't have to be an animal. And it happens when there's a chemical reaction, usually inside of their cells, between two molecules. There's a light-emitting molecule that we call a luciferin. It reacts with oxygen, and that energy from that reaction produces light. But it doesn't happen. It gets accelerated by another molecule that we call a luciferase, which just kind of brings the two together, and it allows them to control when the light turns on and off. So we use these names, luciferin for light emitter, but there's a different luciferin chemically in a firefly than there is in bacteria, than there is in a jellyfish. And so it's just a general term for the light emitting part of the bioluminescent reaction.
Yeah, so that's really interesting that all of these different organisms are capable of bioluminescence. And I guess it must have evolved independently a number of times, because, let's run through a list of organisms. So you said bacteria.
Yeah.
And of course, some mushrooms can do it. Some dinoflagellates, so a type of phytoplankton, unicellular photosynthetic plankton. What else is on your list?
I mean, it's almost easier to say the things that don't bioluminesce.
Okay.
Because in the ocean especially, there are so many things, from jellyfish to shrimp to squid to fish, sea stars, even clams. You know, there's at least a couple bioluminescent relatives in almost every group of marine invertebrates. There's even bioluminescent sharks.
Sharks. Yes, he said sharks right there through that distortion of conducting an interview over Zoom.
There's no bioluminescent mammals, either on land or in the ocean.
Oh, well, that's very boring of us.
Yeah, I know. We think we're so special, but we actually...
Well, we'll have to change that.
One of our biggest shortcomings.
Okay. But are there...
and even other terrestrial vertebrates, and no flowering plants, no like green land plants do it. and no birds, no reptiles, things like that.
Okay. Oh, so there are some things ripe for genetic modification here, I think, because that would be fun.
Yeah.
Glow in the dark birds and glow in the dark flowers. So what's your favorite bioluminescent organism?
That's pretty hard to choose a favorite. I mean, I guess comb jellies, these ctenophores, are definitely one of my favorites. That's one of the main groups of animals that we work on.
In case you're wondering, ctenophores, or the comb jellies as they're also known, look sort of like transparent grapes that swim not by pulsing like jellyfish, but by beating rows of hair-like cilia. And they're very, very cute, and I hope someday you have a chance to see some out in the ocean. And now back to Steve's list of his favorite bioluminescent organisms.
You know, even in a fish, which I'm kind of biased towards invertebrates instead of vertebrates, but there are some really spectacular fish displays that are like blindingly bright, even, you know, to a person when you see them flash, they're just so incredibly bright. Or they have light organs all along their bellies that they look like the running lights of a train or a fancy souped up car.
Oh, that's cool. So have you been in the
squid
deep sea?
have really amazing bioluminescence.
Have you been in the deep sea?
Yes, I've been in the deep sea
and seen this.
I have seen some of it in, you know, with my eye from a submarine, but usually we at the place where I work, we use remotely operated vehicles. So we will usually collect things down as deep as, say, 4,000 meters and then bring it back on the ship into the dark room and kind of explore their bioluminescence in the confines of the dark room. But having said that, we do have a new low light camera that we put on our sub. And so we can actually go down and fly around and film some of the things doing their bioluminescence in their own environment. That's a relatively new development. Like we didn't have good enough cameras until maybe the last 5 years to do that kind of thing. But now we can do it in 4K, full color, low light. And so you can see with the camera what we could see with our eye previously.
So at 4,000 meters, which is very, very deep in the ocean, it's a couple miles, right?
Yeah.
What is bioluminescing and who is the audience?
So unlike in a cave in the deep sea, even though it's still totally dark, their animals don't really lose their eyes. And because of that, I mean, I think that is because there's bioluminescence down there.
Okay.
So it's the way that they can communicate most efficiently and effectively. But at 4,000 meters, there's everything from sea cucumbers to crinoids, which are sea lilies that are also like starfish relatives.
But they don't have eyes.
They don't have eyes. So a lot of the things that emit light are not seeing the light. But there are fish and shrimp and things like that down there that can see the bioluminescence.
So what's in it for a crinoid? Yeah. I thought those were filter feeders.
Yeah, they are. There's a lot of things like there's a lot of octocorals, which are like bamboo corals and deep sea branching corals are super bioluminescent, just incredible. Like sometimes we literally, with the sub, with the ROV, we take a paintbrush and we'll brush it along the branches of these things. And it's like you're painting with light because wherever you brush it, it starts to light up.
Okay. So this is so they don't get eaten. This is to be like, go away. Or
it's presumably, it's either to, I mean, there's a bunch of, we can talk about all the different functions that organisms supposed to use bioluminescence for. In the case of these attached organisms like sea pens and corals and crinoids, it's either thought to be like a burglar alarm, so it attracts a predator of whatever's bothering them. Or my former advisor, Jim Case, thought that it was like apparent motion, where it would seem like something was moving away, but it wasn't actually moving away, but the light was kind of moving. There's also their eyes are like super sensitive and they don't see things very much, the predators. So if you get a big blinding flash, it can be just like us getting a flash bulb going off in our face where you're temporarily blinded or stunned. And there's even some cases in jellies where sort of speculate that the bioluminescence is warning coloration, like in a brightly colored butterfly, for example, you would think that that would attract attention, but it's actually just a warning that I'm not really that good to eat. So for a jellyfish or a coral that also has stinging cells, you could be saying, look, I know you found me, but you don't want to mess with me because you're going to get stung.
Okay. And what about fish? When fish are bioluminescent, what are their reasons?
So fish use it for, I would say, at least 4 different reasons. So one of them is called counterillumination, and we see it in a lot of fish, squid, and shrimp. They'll have light organs just along their belly, even sharks. And they use this as like an invisibility cloak to block out their silhouette from the dim light that's coming from above. So there's a lot of predators down there that have upward directed eyes, or they're just looking up and they're looking for a shadow swimming over their head from the very dim blue light coming from the top. And you can mask yourself from being detected by replacing that light with your own light organs on your bellies. And they go to great lengths to match it. They'll match the intensity, but they'll even match the color. So there's some squid that will shift from blue to green, depending on if they're shallow, counter-illuminating against moonlight, or deeper, counter-illuminating against the more blue daylight.
Oh, that's
one function.
So they have they have different enzymes, not just one type of luciferin.
Right.
Or
how do you do different luciferase?
Mm-hmm.
The luciferase can control it, like in fireflies. There's fireflies that come on at different times in the evening, and they'll have a range from green to yellow. And those are all controlled by the same light emitting molecule, but a different luciferase sequence causes it different colors. In the ocean, the squid are turning on and off different sets of photophores, just like they have different sets of chromatophores for changing their coloration.
Oops. I have to jump in and say that photophores are organs on marine organisms such as fish or cephalopods that produce light, and chromatophores are cells that produce coloration.
And in some cases, the photophores can have little filters over them, like a color filter over it. But actually, I don't know if that's entirely explains it, if they have 2 different luciferases for the different sets of light organs.
No, but really.
So that's just one function for fish.
Mm-hmm.
But they also use it as searchlights to find prey. They have ones forward directed on the front of their head to shine out like little headlights. And in some cases, those are red so that only the fish can see the light and their prey can't see it. Then there's also anglerfish and things that have other lures that they stick out and wiggle around to glow and attract. And then there's ones that have really bright, the ones I was talking about that are blinding, have these light organs that we call stern chasers. And it's these 2 really bright light organs just right in front of the tail fin. And so they'll flash those really quickly and briefly as they dart away. But some actually, there's a 5th one where there's a fish called a tube shoulder that will squirt out bioluminescent cloud to swim away, so it can leave a smoke screen. And that's another pretty common function. But that's just within fish. That's like 5 different reasons. Oh, and attracting mates. So there's some that are thought to school and have species-specific and sex-specific photophore patterns, light-emitting dots on their side, kind of like a constellation of stars that are thought to attract mates. So,
wow. Okay. Who knew the deep sea was such a crazy place?
And that's just what we know. It's really hard to observe all this stuff, as you might guess. So,
well, how do you get it up to the surface and into your laboratory? That must also be a bit of a feat.
Yeah, people always expect things to explode when we bring them up to the surface, but really that is more of a problem in a fish that has a gas-filled swim bladder. So the air will, the gas that's in there will expand as they come to the shallower depths. But most of the things, and even the fish that are down in the deep, don't have any gas space. So they're kind of liquid inside, liquid out. And pressure, until you get down to maybe 3,000 meters, the pressure doesn't seem to be that big of an issue for their viability when we get them up to the surface. So we collect them really gently into these tubes. They're sort of the size of a paint can, and just bring them up and unload them from the ROV and try to keep them as cold as possible
Okay.
while we're processing them.
So is that, I guess you're doing this with robot arms. Is it tricky to screw the lid on a jar with robot arms?
Yeah. Well, so it is like a video game. We literally have like a joystick that's from a fighter plane to control the vehicle going left, right. And then we have one for your left hand that is going up and down. So you do have to fly this huge vehicle around a tiny jelly that may be the size of a grape or something. But the samplers, so you move the sampler over the vehicle, and then there's hydraulics to operate these two sliding lids that close the top and the bottom simultaneously and seal it. So we don't actually have to like have a separate hand to screw the lid on.
Oh, okay.
We use these actuators.
Is your whole little remotely operated vehicle is really just a big sampling device on some level with cameras and lights and all sorts of gizmos, so you know what depth you're at and what temperature and all that kind of stuff.
Exactly.
And then are these?
Yeah, so it can be reconfigured. If you're working on the bottom, then you do pick stuff up with a little claw, and like picking flowers or something and put it into a tray. But when we are working in the water column, between the surface and the seafloor, we use these other sets of samplers like suction samplers and those samplers I was describing with the two lids to collect things that are drifting or swimming in the water column.
How long can you keep these creatures alive in the laboratory?
It really varies. Some of them we can keep alive for weeks or even months, actually.
Oh, that's amazing.
Depending on what it is. And actually, so our sister institution is the Monterey Bay Aquarium down in Monterey. And we work with people there, but they've been able to keep some things that I never thought they would be able to keep, these really fragile jellies and stuff. And they figured out ways, special chambers where they don't bump into the walls and where the water doesn't get all accumulate ammonia, things like that. And so they've been able to keep some really fragile jellies for weeks or months, and even take them through their life cycle, which allows us to do some experiments, even with bioluminescence, to see, 15 generations later, is this organism still bioluminescent, or did it lose its ability to bioluminesce? And so they're able to keep some things for pretty long. But on the other hand, there are some things where we literally have minutes from the time we get to the surface to try to document the jellies, because I think it's largely due to that pressure effect, which we're also studying, where their membranes just don't function unless they're at high pressure. And so their skin will start to like melt, or not their skin, but their tissues will. Oh,
the poor things
start to melt. So I know it's very sad. So
yeah, well, it's weird being a biologist, isn't it? You have to kill what you love.
I know. People always ask, like, do you let them go? Do you, what happens to them after you collect them? And like, I hate to break it to you, but on the other hand, it's so much more selective. It's not really indiscriminate. Like in a lot of cases in the past, you'd trawl a big net through the water and you're just grabbing everything, even a lot of things, most of which you don't want is the non-target organisms. So it's kind of like fishing, or even just driving your car, to work. Anything that splats on your windshield is just like, indiscriminate bycatch. So for us, we have, I think there's 12 of those D samplers, 12 suction samplers, and you can sometimes put two things in a sampler. So we'll go on a dive and we'll select, these are the 40 things that we actually want to bring back and study out of the hundreds or thousands of things that we pass on the way. So
how do you? Okay. So you go with a goal. Otherwise, you'd be like a kid in the candy shop. One of these.
Well, it is still a little bit of the candy shop action, because when you're down there, you never know what you're going to see. So sometimes we see unexpected things or rare things that we haven't been able to study before. But I also, in the past, in bioluminescence research, in the early days, they would collect they collected like 500,000 jellies to extract out the bioluminescent chemicals, purify them enough to like characterize them and study them. But now we can take one jelly and we can sequence its transcriptome or its genome, all the genes. We can extract the proteins and run them on, do chemical characterization of them with mass spec or HPLC or something. So I just, I really think we're amplifying, we're getting the most out of these organisms that we can, even if it's a super fragile, rare one-off. Once we get its genome or its transcriptome, then we can ask all kinds of questions that you wouldn't have been able to ask before.
Oh, that's really, really cool. So what is the weirdest thing you've ever stumbled across down there in the deep sea?
Well, there's been a lot of interesting things. I mean, we found an octopus that was eating jellyfish. It turns out that that whole group preys on jellyfish. We found a
is it weird for an
siphonophore
octopus to eat jellyfish? I've never thought about the dietary habits of octopuses, octopi.
Well,
Of the
I mean, you
octopus.
just think of them as eating crabs, and especially in the deep sea, squids will often eat fish and things like that. So it was, I think, pretty unusual to think about an octopus that subsists on jellies.
Are there many calories in a jellyfish?
Well, there aren't a lot, but I think they make up for it in terms of volume. Like, in the deep sea, you can't really be that picky. So
okay,
if there's a niche, something will end up filling that niche. I mean, some things are eating marine snow, which is basically like snot.
There's all sorts of good stuff in there, though. It's very loaded with carbs. Is some marine snow bioluminescent? It must be. I have to jump in here again and wave the flag for my old friend and former research subject, marine snow, which consists of myriad and sundry biological and mineral particles in the ocean glommed together with mucopolysaccharides, aka snot, and generally sinking impressively quickly from the surface ocean to the deep sea.
It is, but I guess there's 3 types of bioluminescence associated with marine snow. Larvaceans make their own mucus house and they embed bioluminescent particles in them. So these are these little tiny
on
tadpole
purpose
shaped invertebrates. Yeah. So their houses will have bioluminescent inclusions that are just derived from them. And they'll remake a new one of these mucus houses a few times a day, perhaps. And so that when that becomes the nucleus of a marine snow particle, it has bioluminescence. But there's also dinoflagellates
like
that
a
get
comet
accumulated
through the deep sea.
Yeah, it's just sort of, that one would bioluminesce only when you touched it, but ones that are colonized by bacteria will glow more or less continuously.
Mm-hmm.
And a lot of those are derived from fecal pellets. So like fish will have bacteria in their gut, and then their fecal pellets will form a marine snow particle that glows. And then there's sometimes the dinoflagellates that you mentioned earlier, like these surface single-celled algae, photosynthetic protozoans, will oftentimes they'll be accumulated into marine snow. Marine snow just kind of, it literally snowballs as it goes down. And so it's collecting all these other things. I've seen bioluminescent molts of crustaceans. So they have to molt
so they're really
arthropods. Yeah, and so there'll be this like thin shell and it gets colonized by bacteria that are also bioluminescent. So I've seen like glowing molts of marine snow going down there. But yeah, I think another of the weirdest ones is the siphonophore. So these are jellyfish relatives that can grow to be tens of meters long, and they have their own inherent bioluminescence, often that we think is for a defensive function. But this one group has bioluminescent lures that it flicks. So it's like an anglerfish many times over that's attracting other tiny fish in the deep sea. That was one of the fun ones. We found a mammoth tusk, which is not really on topic, but that was one of the more surprising things that we found.
Okay, but not bioluminescent.
No, not that.
But just in the Monterey Bay, or where were you? You just not lying in the sediments?
No, it was offshore. It was on top of a seamount, actually. It was like
that's random.
Kilometers offshore.
But in California.
Off California.
California. Okay. Oh wow, poor thing. I wonder how it lost its tusk and got out there in the middle of the ocean and then hit a seamount instead of going all the way to the bottom. What do you think about potential technological applications of bioluminescence? I know there's a lot of interest in street lights, and there's, I guess, some uses in medicine. And of course, it'd be really cool if you could have bioluminescent clothes or what do you think about any of this?
Well, so there are definitely the medical end of it. There's many, many important applications for bioluminescence and fluorescence in medicine. And that for sure is something that I think is promising to sort of invest your time and effort in. And I don't want to be a downer, but I think the streetlight idea is kind of a non-starter to some extent, because for it to be actually bioluminescence is actually really dim relative to even like an LED light or something like that. And so you would need a lot of chemicals that have to be somehow synthesized to produce enough quantities to make visible light. At this point, that is pretty expensive. So even if you had a culture, say, of algae that would photosynthesize during the day, I actually have some in the room here with me. Let me just keep them on a timer on a light. So the algae at night, they'll make some amount of light, but they'll relatively quickly be exhausted. If you shake up this culture of dinoflagellates, you may get 5 minutes of fun out of it.
I actually had one of those about 20 years ago. Noctiluca, I think it was. And yeah, you could shake it a little bit and it would glow for a few seconds, but then you'd have to wait if you wanted to see it again. You could definitely not read by it. And then a visitor shook it way too hard, and that was the end. That was the end of those poor little dinoflagellates. There was way too much turbulence for them.
It's beautiful to see, but I think functionally, the amount of energy that you're putting into growing the culture during the day is way more than you would get out from them at night. So why not just have that LED be your source of a street light at night?
Because it's cool. Because it would be really cool. But I mean, I hadn't thought of it.
It's dim.
I hadn't thought of doing it by growing dinoflagellate cultures in streetlights. So you would actually have a bioluminescent organism, and then I guess you'd have to slowly flip the light over or shake it back and forth. That's interesting. Because I was trying to think, how could you just have the enzyme and the chemicals there and get them to react and regenerate? But yeah, of course, if you just had an organism doing it, then a photosynthetic organism, you just feed it some nutrients every now and then. But it's not very bright.
Well, so I think the more promising might be bacterial or fungal. So mushrooms and bacteria will glow pretty much constantly if there's oxygen there. And so those, I think, would be a little bit more promising for just like ambient space, like architectural applications. But again, they're dim. So the bacteria wouldn't need light, dark cycles to grow, but they'll need nutrients and, some kind of environmental control.
What kind of bacteria are bioluminescent? I have to say, I don't know.
Vibrio. There's a thing called Photobacterium and Vibrio. So it's related to Vibrio cholera. Yeah. But you can grow them just in a broth, LB or whatever. You can grow them in a nutrient rich broth. So they could potentially, go through the night or whatever, but they'd still be relatively dim. Like you said, you would hardly be able to read by them. The fungal one is actually pretty interesting because Ilya Yampolsky, some Russian scientists, figured out the fungal bioluminescence chemistry. And the problem with a lot of these is that we have the luciferases cloned, but we don't have the biosynthetic pathway for the luciferins cloned. So if you make a transgenic organism, you put the luciferase in there from a jellyfish or photoprotein from a jellyfish, you have to provide the light emitting substrate somehow exogenously. So you can synthesize it in like an organic chemistry setup. You can create your own light emitting molecule, but you have to constantly provide that in order to actually get light out. So one of the things we're working on is what is the genetic pathway that organisms are using to make that molecule, and can we replicate that in some kind of a genetic cassette that would let you make bioluminescent things based on the jellyfish light emitter.
Okay.
So that's the negative case where we don't yet know how to do that. But in the other case, Ilya and his group figured out the fungal luciferin and luciferase system. So they have the way to make the light emitter and the control system. And what's really cool about that is that the required substrate for that luciferin is in plants. Okay. So they've taken the fungal system and put it into plants and actually made glowing flowers and glowing plants. And I think that to me is the most promising thing that you could, have for an industrial application, because you don't have to provide this expensive organic molecule to get the light out. The plants are going to be making it themselves.
Okay.
And so they have a company that actually is commercializing it, but it's still really, really dim.
Okay. And it was just, what's their angle on that? Just people would buy it because it's cool, or?
I think there is, to some extent, that there's been talk of like, oh, well, we could have crops that tell you when they need water or when they're being attacked by insects. Okay. So they're like self-reporting their status through light. I think they have a bunch of angles that they're trying to do.
So it's not all just
that one. I think, I would keep an eye on that one. But I think like there's a town in France that is prototyping some bioluminescent like street signs and stuff like that. But I don't think it's all that promising, to tell you the truth. So
what's in it for a glow in the dark mushroom? Is it just saying don't step on me, or what's it doing?
I think in that case, it's thought, so glows in general, glows are thought to be attractive and flashes are thought to be repellent.
Okay.
So, in an otherwise dark environment like the deep sea or a forest floor at night, things are going to be attracted. They're going to see something glowing and they're going to go over to investigate what it is. So in the case of a mushroom, it's thought that insects will come trundling along. They may nibble some of it, but then they'll get spores on them. And as they go off, they'll distribute the spores.
Okay.
It's been, at least, one of the hypotheses that I've heard is that it's to help it propagate itself.
It's amazing that there are so many things that you can do with bioluminescence if you're out there in the wild.
It's just a really efficient way for something to communicate. Like if you think about one of these dinoflagellates, it's less than a millimeter long, right? So if you imagine a millimeter size organism sitting on your desktop and it wants to send you a signal, like in one of those sci-fi movies where they're like, the people are yelling out, but they can't hear them and the cat's coming to eat them or whatever. But so if you imagine that 1 millimeter thing sitting on your desk, it can't communicate through sound, vibration, chemicals. There's really no other way. But if it was dark and it made a little flash of light, you would see it, and you can see it from individual dinoflagellates. So
really? Oh, yeah. Wow.
Yeah. Like the specks, when you shake up those jars, you see all the little specks.
No, I remember the first cruise I was on during my life as an oceanographer. I had been working on the back deck of, what ship was that? The Sproul, a tiny, tiny little ship. And it had been, the whole back deck had been awash. And I had been helping collect the zooplankton in the nets. And then I got back to the cabin, where there's 6 people in the cabin, so you couldn't turn the lights on. And then I was trying to take my wet shoes off and they were flashing at me. And I realized, oh my gosh, awesome. I'm torturing little dinoflagellates here. Yeah, that's right.
They're communicating to you over a great distance, many, many body lengths, and, to something many, many orders of magnitude larger than them. And in the ocean, it's, especially in the deep, it's clear enough and the eyes are sensitive enough that people have calculated that they can probably function over like 50 or 100 meters.
Wow.
Bioluminescent signal can be
water.
seen. So
that's extraordinary.
Yeah. So it's just a really effective way to communicate. And it's probably, the most common form of communication on the planet when you think about it.
So what are dinoflagellates saying to each other?
So dinoflagellates go back to 2 of the functions that we discussed. One is thought to be directly deterring predators by like flashing in their face right when they're about to be eaten. And some people did some experiments a while ago to show that seemed to be happening. But I think even more compelling is the burglar alarm. And there have been some experiments done on this too, where you have a tank with dinoflagellates in it. And so it ends up being like a minefield. So an organism swimming through there leaves a little bioluminescent trail behind it. And then other predators come and eat that. So, if you're a dinoflagellate and something is grazing on you, you may not make it, but the rest of you in the surroundings, by this group effort of highlighting the predator, the grazer will remove it from the ecosystem.
Dinoflagellates are so wicked. Wicked little plankton. Well, and
they're just defending
themselves. Didn't they take out a submarine? There's this story about a submarine in the deep, and it was discovered by another submarine or somebody, or maybe it was an airplane trying to bomb it or whatever, because it just left this huge bioluminescent wake.
Yeah, in the 80s, I think even back in the 70s, 80s, 90s, the Navy would fund a lot of bioluminescence research because they wanted to understand it. And frankly, my advisor was asked, how much poison would it take to turn the ocean black surrounding, our ships so that they would not be bioluminescent? But
so
they're worried about
answer?
Even like, basically it's not possible. I think he was just like,
what a terrible thing to do.
At the naivety. I know. But
it is a bummer,
even for like Navy SEAL operations and stuff like that, like individual people or scuba divers up to like a nuclear sub or something. I think it can essentially reveal the location the same way it does in nature.
That's true. I've been night diving and you feel like Tinkerbell. Mm-hmm. Little sparkles that you totally. And you do feel like you're sort of saying, hello, come eat me, I'm here. Before I say thank you, do you have any parting remarks you'd like to make about bioluminescence or the deep sea? Or
I guess I think I would like people to appreciate that it's really really common. Like, it is this magical thing, but it's also, there's about 3/4 of the animals that we see with our ROV, all the way from surface to 4,000 meters, 3/4 of them are able to produce bioluminescence. Wow. So it's really common and diverse. Everything, from single-celled organisms to starfish, and pretty much everything in between in the ocean has some members that can make light. So just appreciate that it's out there, and it's not an anomaly.
Oh, cool. Okay. No, it's a really magical world out there, and we should remember that sometimes. So, okay. Well, thank you, Steve, very much for your time. I appreciate that you're a busy guy running a big lab, and enjoy your further explorations of the deep sea and of bioluminescent organisms.
Thanks, Christina. It's great to chat with you, and thanks for the interest in my favorite topic.
Thank you for listening to Solarpunk Presents, a series embedded within the Solarpunk Futures podcast, written, hosted, and produced by Christina de la Rocha and Ariel Kroon. This podcast is a part of Solarpunk magazine, which is published by Android Press, which is located on Kalapuya Ilihi, the traditional indigenous homeland of the Kalapuya people. Today, descendants are citizens of the Confederated Tribes of Grand Ronde Community of Oregon and the Confederated Tribes of the Siletz Indians of Oregon. The opening and closing music for Solarpunk Presents is Water Cooler Gang by Monkey Warhol and is available for use under the Creative Commons Attribution 4.0 International License. So, thank you again for listening, and until the next episode, stay Solarpunk.